Hello, everyone. I welcome the students, researchers, scholars, teachers, and scientists all over the world. This is Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, today I'm going to share my information, my knowledge, and my experience under the title of the Carriers, Advantages, and Applications of Immobilized Enzyme. For this purpose, I have collected the information from different scientific data sources like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, MBAS, directly of Open Access Journal, ProQuest, CNKI, that is China-based database company, stand for China National Knowledge Infrastructure. Some information collected from Crossref and ResearchGate. Before moving forward, I would like to talk about content and coverage. I'm going to talk about what is an immobilized enzyme, what is the enzyme immobilization, then the carrier advantages and the application of immobilized enzyme. Okay, well, an immobilized enzyme is an enzyme attached to an inert insoluble material, most of the time the calcium alginate. Immobilized enzyme produced by reacting a mixture of the sodium alginate solution and enzyme solution with the calcium chloride. Immobilized enzyme can provide a resistance to change condition like change in pH or the temperature. Then immobilized enzyme retain the catalytic activity and can be used repeatedly and continuously. It is produced of confining the enzyme molecule to a solid spot over which a substrate is passed and converted into the desired chemical or the product in the pharmaceutical and health industry. Okay, well, there are three main type of the carriers that are used in immobilized enzyme or immobilization of the enzyme. The first one is the natural, then synthetic, then inorganic. Natural is divided into the three further categories: structural proteins, for example, collagen, then globular protein, for example, albumin, the carbohydrates, an example of carbohydrates are the dextran. Synthetic organic, example, polyvinyl epoxide. Then inorganic, example of inorganic are the glass, silica gel, and bentonite. Okay, well, typical model of the bioreactors and the mode of immobilization, it comprises of the two main parts. The first one is the bioreactors used for immobilized enzyme, and the second part or the lower component of this figure comprised of the mode of enzyme immobilization used for respective bioreactors. The bioreactors are four main types. The first one is back bed reactor, then fluidized bed reactor, then retentive state, and the final fourth one is the membrane reactors. And we have five different kind of enzyme immobilization used for respective bioreactor. The first one is the cross-link enzyme aggregate. Second one is the immobilization on the solid spot. Then entrapment in the matrix or the gel. Then permeabilized cell or the semi-permeable membrane. And the last fifth one is the lactate immobilized membrane. These are the five different kind of the mode of the enzyme immobilization used for the respective bio reactors. Okay, well, now I would like to talk a little bit about the advantage of immobilization, protection from the degradation and the deactivation. Immobilized enzyme used for the reuse of the enzyme for many reaction cycles, lowering the total production cost of enzyme mediated reaction. It is also used to increase the ability to stop the reaction rapidly by removing the enzyme from the reaction solution. It also enhances the stability. It is also used for the easy separation of the enzyme from the product. There are the different application of immobilized enzyme in health, biomedical, and health science. Immobilized enzyme are used in the medicine since 1990. Immobilized enzyme are used for the diagnostic treatment of different kind of the diseases in medical and health science. The inborn metabolic deficiency can be overcome by replacing the encapsulate enzyme. For example, enzyme encapsulated by the erythrocyte instead of the waste metabolites. Okay, well, the enzyme encapsulation through the electroporation is the easiest way of immobilization in the biomedical field, and it is a reversible process for which enzyme can be re- regenerated. Okay, well, immobilized enzyme also used to produce or synthesize different kind of the medication like penicillin amylase used for production of the penicillin, G-amoxicillin, macrolide, antibiotic, nicotine can be produced by the living cell of Streptomyces species. Steroid amino acids, beta tyrosine, and the levodopa are produced through the immobilized enzymes. These are used for the diagnostic purpose too. The glucose biosensors used to identify the glucose level in patient's blood. The glucose biosensor used for the following purpose, like it contain a layer of immobilized glucose oxidase enzyme bind with any glucose in blood which is oxidized with the dissolved oxygen from the solution to the hydrogen peroxide and the gluconic acid. And electrolyte, most of the time with platinum, used to measure the oxygen concentration to make the hydrogen peroxide. The size of electrical signal is proportional to the concentration of the glucose in the blood. Okay, well, this is the utilization of the immobilized enzyme in the food industry. There are the three main processes where immobilized enzyme used for the production and synthesis of the food: organic, inorganic composite, and the inorganic material. And third one, the organic polymerase. There are five different kind of the enzyme involved sufficiently used in food industry: pectinase, lipase, invertase, lactase, and amylase. Okay, well, in conclusion, the immobilized enzyme performance is directly related to the substrate provided condition, immobilization method, and carrier type, because carrier increase the stability and compatibility under challenging condition. So the scientific development has a vital role to use the resources in more efficient manner to produce the quality dairy, pharmaceutical, chemical product in best possible fashion. Okay, well, before closing the session today, I would like to gratitude and greet to my colleague, my coworker, my friends, Sherry Baskat, Wolfgang, Daniel, Dustin Hacker, Marwa Al Maghribi, then Robert Heyman, Kunza Noreen, Faisal, Tarimil, Ishtiaq, Arslan, and Amin. These are my people that they help me a lot to complete this project and make it fruitfully conclusive. I also acknowledge the kind help and support of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, uh, I appreciate you for watching the video and staying with me for so long time. I hope see you soon in my next video, and take care. Bye for now.